Alrighty guys, welcome back to your remote learning experience. This lesson is for Thursday, April 16th, and I hope you guys had a great Wednesday, and hope you had a good time. We're able to get outside. It's been really beautiful this week, so definitely take advantage of that nice weather. So we are going to pick up today where we left off on Wednesday. Okay, this is a little bit confusing because I filmed these the day before, so I get confused with all the dates. But Wednesday's lesson was to watch the video on the very first two steps of DNA replication, and today we are going to wrap up the steps of DNA replication. There isn't going to be an assessment on DNA replication. Okay, it's kind of like a little extra kind of content that we're doing right before we get into genetics. But I thought it'd be a good idea to have a better understanding of DNA and how DNA works. Okay, so I'm going to get into our whiteboard from yesterday just to kind of wrap up where we left off as soon as this whiteboard loads for me. Usually it takes a while. All right, so yesterday's step was that primase, which is an enzyme, makes an RNA primer, okay? And these RNA primers are kind of these black blocks right here, and they have bases attached to them. And remember, the RNA bases are A, C, G, and U rather than A, C, G, and T. Okay, so everywhere that you see a green kind of base right here, that is going, right here, getting my colors mixed up, that is going to be a U for RNA instead of T, okay? So these primers are made to help DNA polymerase know which direction to go in, okay? Because DNA polymerase is very specific and can only go in one direction, and that is from the 5' to 3' direction. Okay, we talked about the directionality of DNA and how it has kind of like north and south and east and west. It's just 3' and 5'. Those are the directions that DNA knows. Okay, so I want to move on to our third step of DNA replication, which I believe is this one. Okay, so the third step of DNA replication, I'm actually going to be breaking down into two parts because it can get a little bit complicated. But essentially, the third step of DNA replication is that DNA polymerase, which is our builder enzyme, begins to build the new strands of DNA. And it does this by adding complementary base pairs. So, in the very first step of DNA replication, we split this piece of DNA right down the middle, okay? And so now the DNA strands are unpaired. So what DNA polymerase is going to do is add bases to this unpaired strand of DNA, okay? So it's beginning to build a new strand. So up top, wherever there is an A, the DNA polymerase is going to add a T. And whenever there's a C, the DNA polymerase is going to add a G. Okay, so I'm going to see if I can hopefully remember. I think this one was T. Black was C, I think. And blue was G. Okay, so whenever there's a red A right here, a red base, that's going to be adenine, and so DNA polymerase is going to add T, so that A will bind to T. Okay, so this is DNA polymerase right here. It's just a blue circle, okay, and it's going to build in a specific direction. So on this top strand right here, DNA polymerase is going to be working this way, okay? So instead of me saying that DNA polymerase is working from right to left, okay, because it's starting here and going towards the left, I'm going to say that DNA polymerase works from 5' to 3'. Okay, so I'm going to get into the directionality a little bit because it can be pretty confusing. Okay, but if you've noticed these two strands, okay, there's a blue strand right here on the bottom and a green strand right here on the top. And remember, DNA goes in the strands go in opposite directions. Okay, so if you see in your DNA models that you've made, you'll see that one of your DNA strands goes up and then the other DNA strand is kind of read upside down. Okay, so actually, let me see if I can get an image of that real quick just to show you. DNA paper model. Okay. So this is what your model was supposed to look like. So if you see one of the strands of DNA is going up like this, so you can actually read the words on the particular structures. But on this side, it's going upside down. Okay, so deoxyribose sugar is upside down, so is phosphate, and so are the bases. Okay, so DNA kind of goes in opposite directions on each of the strands that are in DNA. Okay. So one of the strands will go from 5' to 3'. And if this top strand is going 5' to 3', that means this bottom one has to go in the opposite direction. So this one goes from 3' to 5'. Okay, so the top one, once again, 5' to 3'. The bottom one is 3' to 5'. Okay, but DNA polymerase can only work from 5' to 3'. Okay, a common misconception I've seen in the past is that, oh, since DNA polymerase builds from 5' to 3', it should go in this direction on this strand because this is 5' right here and this is 3' right here. Okay, but you have to think of DNA polymerase in which way it's going to build the bottom strand. Okay, so eventually I'm going to have a new strand on the bottom of this top strand, okay? This top strand right now is 5' to 3', okay? And this bottom strand is going to be 3' to 5', okay? So if I want to get this bottom strand, if I want to add this bottom strand, DNA polymerase is going to have to work this way. Okay, because think of DNA polymerase building in what direction the bottom strand is going, not the top strand. Okay, so the new bottom strand of this green strand right here is going to go from 5' to 3'. So DNA polymerase is just going to work its way down this way. Okay, so it's going to be a nice and easy route. Okay, and that's a little bit different with this bottom blue strand. But this top blue strand is actually what we call the leading strand. And all that means is that DNA polymerase has it pretty easy right here. It can just go along in the direction that it wants to go, okay? It can just build continuously. Okay, so I'm going to write that down. DNA polymerase builds continuously. Okay, so that can be a little bit confusing, but I am going to attach an Amoeba Sisters video and they do a very good job of explaining the directionality of DNA and what happens when DNA is copied. Okay, so that is how the leading strand or this top green strand is made. So now I'm going to move on to talking about the bottom strand because the top and the bottom strand in our example here are actually made differently. Okay, so this is still step 3 because DNA polymerase is building new DNA strands, okay? But this time we're going to focus on this bottom blue strand right here, okay? So if you look at this top green strand, as you can see, DNA polymerase has been doing its job and adding those DNA bases onto that green strand right here, okay? So eventually, this is going to be a completely new DNA strand. All right, so I added that DNA polymerase has built some of the strand and it's going to continue to build, okay, until it's done with the full strand of DNA, okay? But this strand, this bottom blue strand, works a little bit differently with DNA polymerase, okay? And that's because DNA polymerase can only work from 5' to 3', okay? So with this bottom strand right here, I'm going to kind of do a mini version of it. It's initially going from 3' to 5'. But remember, when we talk about which way DNA polymerase is building, we want to think about the strand that it's going to build, not the strand that's already there. Okay, so it's going to build a new green strand, and that new green strand is going to go from 5' to 3'. Okay, so in this bottom strand, DNA polymerase can only work this way. Okay, and that's not as simple, okay, because DNA polymerase is going to have to work backwards in this case, okay, because if it tried to go from 3' to 5', it can't work like that. Okay, so that is not going to happen. We want it to go from 5' to 3', okay? And in order to go from 3' or 5' to 3' on this bottom strand, DNA polymerase is going to have to work backwards, okay? So initially, what DNA polymerase is going to do is it's going to start here with this RNA primer, okay? And it's going to lay down some DNA bases just like this, okay? So there's going to be a green one right here. There's going to be a red one. There's going to be black right here. Okay, so DNA polymerase just built this strand, okay? But now what DNA polymerase is going to have to do is go back to this RNA primer and then do the same thing again, okay? So I'm going to draw another DNA polymerase, and it is going to add more DNA bases. Okay, so as you can see, DNA polymerase on this bottom strand works in sections, okay? So it can't just move continuously all at once like it does in the leading strand or that top strand. It has to work in sections like this. Okay, so there's a section right here and there's a section right here. Okay, so DNA polymerase will start right here and then it'll work backwards and go right here and then build, okay? So it starts at a point, goes backwards, does that section, goes backwards, does that section, kind of. Okay, so that can be a little bit confusing, so I do encourage you to watch the Amoeba Sisters video because I do think that they do a great job explaining it if my explanations aren't that great. But you can also look in your textbook. I might attach the book pages so that you can take a look, and if you're more into reading things rather than visualizing things, you can absolutely use that as a resource. Okay? So that's why we actually need multiple RNA primers on this bottom strand, is because DNA polymerase has to work in sections, okay? On the top strand, or the leading strand, it just needs one primer because it's only doing one section and it's going to do that section continuously, okay? But this bottom strand, it works in sections. It needs multiple RNA primers. Okay? Oh my goodness, this lags so bad sometimes. Okay, so since this bottom strand has to work in sections and isn't continuous, we actually call it the lagging strand because there's a little bit of lag to it. DNA polymerase has to really work to try to build that piece of DNA. Okay, so this would be the process for how to build this bottom strand or the lagging strand. Okay, and just to be clear, either one of these could have been the leading or the lagging strand. I just decided to make the directions so that this top strand was leading and this bottom strand is lagging. But in any strand of DNA, it could be the top strand that's the lagging strand and the bottom strand that's the leading strand. Okay, this is just the way that these strands are oriented. It's just how I chose to show you guys. Okay, so I'm going to move on to the very last step of DNA replication, which I believe is this one. All right, so the last strand or the last part of DNA replication is that DNA ligase glues DNA together. Okay, so as you can see, DNA polymerase has kind of finished its job and it has built this top strand and it has also built this bottom strand. But you'll notice that this top strand has a continuous sugar and phosphate backbone right here in all the bases, but this one has been broken up into different segments, okay? The name of these segments, these are actually called Okazaki fragments. So that's a term you'll see in your book and also in the Amoeba Sisters video. But the name of the fragments aren't super important. I don't think it was just named after the guy who discovered them. But that is their name, but the lagging strand will have different fragments, okay? And you can also see that I've labeled the directions for the new DNA strands, okay? Because eventually these two are going to be separate. So this is going to be its own DNA strand and this is going to be its own DNA strand, okay? So DNA ligase, I'm going to draw it as like a little square. There we go. All right, and DNA ligase is going to glue these fragments together. So it's going to come in and make sure that this is glued together, and then it's going to go over here and make sure that this is complete and glued together. So after DNA ligase kind of does its job, you will have those two new DNA strands. DNA ligase also helps to make sure that all of these bases are nice and secure and connected together again because they were separated during DNA replication, but now they've been bonded back together. Okay, so that's DNA ligase. It's pretty simple, but also very important. All right. So the last part that I want to talk about for today's lesson is kind of the results of DNA replication. So I want to say that it's this one. Yes, so the results of DNA replication. After DNA replication is all said and done, what we have is two identical sets of DNA. Okay, so we have copies of that DNA. So we have one copy right here. I'm not going to draw the inside of the DNA just for the sake of time, but just know that there are DNA bases in the center of these. Okay, so those are identical copies of DNA, okay, and they are going to be passed on to their appropriate cells during cell division. Okay, so this is why this process is so important. It's because the DNA provides the instructions for the cell and tells everything in the cell what it needs to do. Okay, so we need this DNA to be properly copied in order to pass it on to the cells that are formed during cell division, okay? But it is important to note that there are mistakes in this process, okay? So DNA polymerase is a very intricate and amazing enzyme, and it actually has like proofreading abilities. So imagine that you wrote an essay and you need like your mom or your dad or your friend to proofread your essay. your mom or your dad or your friend would be like DNA polymerase. DNA polymerase can actually go through and make sure that all the bases are paired correctly. However, DNA polymerase isn't perfect, just like no one else is perfect, and there can be mistakes in this process when DNA is copied. Um, Sometimes these mistakes don't really result in anything, but sometimes they do. It really just depends. But we'll kind of, I hope to get into more of this when we talk about genetics. Hang on, I think I have to sneeze. Excuse me. So, we'll get into this when we talk about genetics, about the potential mistakes that happen during DNA replication. But DNA polymerase still is doing a fantastic job because your DNA is actually super long and very intricate. So, the fact that DNA polymerase can add all those bases is actually pretty amazing. Okay, so that is going to do it for DNA replication. I think it is a super cool process and amazing to think about how all of that stuff just goes inside our bodies without us even having to think about it. So yeah, it's pretty cool. So that's going to do it for today's lesson. I hope you guys have a fantastic rest of your day. Feel free to email me with any questions. And yeah, hopefully you get outside today and stay healthy and safe.